This is the long-awaited lecture on the electron transport chain and chemiosmosis. So I want to establish some things about the hydrogen ion that we talked about in our last video when we were referring to NADH. Here we see a mountaintop, snow-capped mountaintop. We have hydrogen at the top and oxygen at the bottom. Hydrogen has an electron, oxygen has electrons, but oxygen keeps those electrons bound super close to itself because it's super electronegative. To release or pull that electron away from oxygen would be like rolling this electron uphill. Whereas the hydrogen ion, this electron is not as closely bound to the hydrogen atom. And it's much easier to liberate that electron from the hydrogen ion. And in doing so, it releases energy. So as that electron is pulled away downhill from this hilltop energy, if you will, energy is released. So that is the value in the electrons in the hydrogen ion that NADH shuttles to the electron transport chain. And that's what we see right here. So this organelle, this entire organelle, is known as the mitochondria. This black line on the periphery represents the outer mitochondrial membrane. The black line right here represents the inner mitochondrial membrane. The space between these two membranes is the intermembrane space, and the space in the middle is known as the mitochondrial matrix. Each one of these colored structures is a protein that make up the electron transport chain. And these proteins are very electronegative. As a matter of fact, as we proceed from left to right, they become more and more electronegative. So the blue one is more electronegative than the green one. The red is more electronegative than the blue, and the yellow is more electronegative than the red. So now we have our NADH delivering those electrons to the electron transport chain. We have these two electrons. Keep in mind, NADH was carrying this hydrogen ion and these two electrons. Those electrons are going to bind to this first protein and the electron transport chain. And what I'm showing right here is just this snow-capped mountain peak. And I'm showing that up right here. I realize it looks like an A, but it's supposed to be that snow-capped peak with this electron at the top. So now the blue protein is going to pull that electron towards itself, and in doing so, it's going to release energy. And now we see here that electron is falling down its gradient, releasing energy in the process, moves down to the next protein in the electron transport chain, and that electron keeps falling to the more electronegative proteins until it gets to the most electronegative thing of all, and that's oxygen. And that's exactly why we breathe in oxygen. So oxygen can serve this role in the electron transport chain. That is to say, it's the ultimate reason why these electrons are jumping from one protein to the next, terminating at this oxygen atom. So one might ask, why are we doing this? Well, the ultimate goal is to push the hydrogen ion into the intermembrane space where there's a higher concentration of the hydrogen ion. As we see here, there's a low concentration of the hydrogen ion in the matrix, a much higher concentration of the hydrogen ion in the intermembrane space. If we want to move the hydrogen ion from the matrix to the intermembrane space, that is going to require energy because we're moving against its chemical gradient. And that energy comes from those electrons jumping or moving from one protein to the next, and in the process, releasing energy that is harnessed to move the hydrogen ion against its gradient into the intermembrane space. Now that we have a ton of hydrogen ions in the intermembrane space, what does the hydrogen ion want to do? If you said move back into the matrix, you're exactly right. The hydrogen ion wants to move into the matrix down its chemical gradient. But to do so, because it is an ion, because it is charged, it requires a channel, which we see right here. And this channel is known as ATP synthase. As the hydrogen ion moves through that channel, the ATP synthase, it's moving down its gradient, it's creating kinetic energy, and that kinetic energy is harnessed to synthesize or produce ATP molecules. In this process, from one glucose molecule and all of those, the 8 NADH molecules and the 2 FADH2 molecules will produce upwards of 34 ATP. Keep in mind we had 2 ATP from glycolysis, 2 ATP from the Krebs cycle, 34 from the electron transport chain in chemiosmosis. If I do my math correctly, I think that's 38 ATP molecules. That is cellular respiration.